So then today we're gonna go more into epidemiology, and then maybe start food microbiology, I think. The goal is so when you look at the syllabus, we're doing epidemiology, the food microbiology is next, and then industrial and applied micro. So my goal is to get you exposure to all the topics on the syllabus there toward the end. I'll just shorten up what I have done in the past since we're a little behind schedule. And then since the exam is, I'll probably hold review session for the final exam on next Wednesday. I'll probably do that. That would give you a whole week then. And so by next Wednesday, I'm hoping to have all the grading done of this last exam. And because I need to think about if I'm going to ask any cumulative questions, and you need to, like, I'll give you that in advance, the topic, so that if I can do that by next Wednesday, that would give you a week. Okay? All right. I think we left off here looking at the idea of when pathogens are interacting with potential hosts, there's usually, especially with viruses, but also with some bacteria and fungi, there's usually this idea of coevolution that will occur over time. And with viruses, that can actually happen fairly quickly. We did see that the COVID virus, for example, did go through a coevolutionary process with the human host. And now I think the last I heard is they're saying it's going to be something that is persisting with us and interacting with us, kind of like the flu virus, all that has persisted with us. And so when you think about host-to-host transmission in particular, so those pathogens that really need a host to be up and moving around so that they can do, from their goal is to spread to as many people as possible, make as many of themselves as possible. And you can't do that if the host is bedridden or dead. It's much harder. On the flip side, if there is no selection pressure to co-evolve. So with the virus should co-evolve to be more mild. And then with the host, it should co-evolve to become more resistant. If there's no selection pressure to have that process going, then things can get really nasty. So now we're thinking about the type of pathogen that really doesn't need a host to fulfill its life cycle, to be able to continue on into the next generation. So probably the classic example of that would be, let me see if I can pull up the table. Yeah, here. The classic example for that idea would be the botulism idea caused by Clostridium botulinum. By the way, I think Monday I made a mistake. I think I was mentioning, what was I saying? I mentioned, instead of, I think I mentioned botulism in Monday's lecture as an example of, and I can't remember what, I'm sorry, I'm very, very tired. And I can't remember what it was exactly, but I remember getting back to my office shortly after Monday's lecture going, ooh, no, that was what I meant to say was the anthrax. Okay, so I think in Monday's lecture, I talked about how you can get botulism from the soil, which is true. And then I said, and there's different versions. That was when I started flipping in my head, thinking about anthrax. So one day, it is actually fairly common for farmers to get and then I said botulism. What I meant to say was anthrax. There's different forms of anthrax. There's a cutaneous form of anthrax that's usually not deadly. And if farmers are working with cattle or swine or goats, sheep, horses, soil, planting seeds or harvesting plants, you can get, typically it's cutaneous anthrax if you get exposed to anthrax. And that is usually not a problem to get treated. Anyway, I just wanted to clarify that I misspoke on Monday. So now going back to this idea, if the pathogen doesn't need the host to move around, spread, if it has another way to stay alive, another way independent from the host that it can live its life quite nicely, then it could kill the host during exposure and never get less food. So Clostridium botulinum, and see the reservoir for that is the soil. So what that means is that it's happy. It's happy in the soil, so to speak. It can live its full life in the soil and be highly successful. It doesn't need a host in order to, be successful. So then if you get exposed to it, there's no coagulation, there's no selection pressure for, in this case, Clostridium to become less virulent. And so that's when you start running into trouble, is that those types of pathogens really, they're not going to adapt in any way. So think about that idea of selection pressure. What is the reservoir, and whether or not, on these other ones, you can see humans are the reservoir. So in those cases, and I think in that TED Talk I'm asking you to watch, he looks at cholera and how it's transmitted via water, but humans are the reservoir. And so that's a really good example, if you watch that TED Talk, of ways to use the idea of coevolution and selection pressure in order to make cholera more mild, with actually some pretty simple ideas that he proposes to put in place. Could go a long way to really making that less deadly. Okay. So often, there's many examples of vector-borne pathogens that can spread like via an insect of some sort. They tend to not really co-evolve with the host. They tend to stay highly virulent. Contact transmission, that would be the case of like if you touch the soil where Clostridium might be located. Again, it's they don't have to survive. They don't have to depend on the host to survive. I'm sure you've heard of herd immunity by now, especially with the pandemic. This idea that how many people need to be immune in order to protect those that are still susceptible. And this herd immunity can be very powerful. It is, but it varies. So depending on the pathogen, this herd immunity, you might need like a whole bunch of the herd to be vaccinated and immune in order for this to be successful. With other diseases, you don't need as high of a percentage. You've probably noticed over just your short life so far that they tend to highly publicize and really push flu vaccinations in the fall. And the reason for that is it can take a lot. You have to vaccinate a lot of people to get to the herd immunity idea. If you can provide herd immunity, essentially the benefit to that is you're breaking the cycle of the pathogen hopping person to person. So if people who aren't vaccinated are in the mix of those that are vaccinated, they're far less likely to get infected because of that buffering effect of all the other people around them that have been vaccinated. So like I said, it depends on the pathogen. For herd immunity to occur for polio, it's 70% of the population in that area, whatever geographic region you're looking at. 70% have to be immunized or immune, and 90 to 95% for the flu. So that's why they have pretty strong ad campaigns and really push it, and your doctor will often offer it. You don't ask for it. They'll just say, hey, you want a flu vaccine? Because of this idea. And so it varies from pathogen to pathogen, how this will work and play out. And they, they don't know until the pathogen's been studied well enough to know, what that percent takes a little bit. But this is a common diagram they give for this idea of herd immunity, with the red being affected. Many people immune and breaking that cycle of host to host. So good strategy. If you choose to, once you graduate, or maybe even now, if you choose to travel anywhere from where you currently are, so if you're in the UP, one of the things that you should do to prepare, and this is no joke, you should be looking at, okay, where do you plan to travel, and think a little bit about that area in terms of the infectious disease profile. This is just one example showing Africa versus the Americas, and you can see by the pie chart very different scenarios. Here in the Americas, infections are only at 10%. Look at what they are in Africa, 56%. On the other hand, look at our cardiovascular disease. We're way higher at cardiovascular disease. So that big green piece of the pie is largely due to the Western diet, the SAD diet. That's why you see it shift there, as well as cancer. A lot of what we choose to eat can either suppress or promote cancer, certainly diabetes as well. But for this topic, what I really want to think about is purple, the light purple, this idea of infections. And so when you go and commingle with other people outside of your normal area, it's always good to have a little bit of a feel for what's going on in that area before you travel there. Here is a map showing emerging and re-emerging infectious diseases. It's difficult. So this is covering viruses as well as bacteria, fungi, and a variety of different mechanisms of transfer. Some of these are host to host, some are vector borne. So it runs the whole gamut of methods of transmission. Yeah. This is one that is gaining a lot of scrutiny. Multi-drug resistant tuberculosis. So multi-drug means many antibiotics are useless against it now. And there was a case study, I have a documentary of a case study where they were tracking tuberculosis from prisons in Russia as inmates got released from the Russian prisons, and then they started commingling back into their, their hometown. And then they were able to track that that particular strain made it all the way from the Russian prison to the Russian hometown to New York City in 10 days or something like that. It was amazingly fast how quick it transferred. Even though that person, it wasn't the person itself, the person released from prison, he didn't travel. It was who he commingled with, and then they were traveling, and they brought that over. So with the ease of travel, the quickness of travel, you look at this map and you might say, well, go around me, but unfortunately we can't be that cavalier about it anymore. So various activities, human activities and concepts that have come about over the last couple decades have created situations that allow for pathogens to spread more easily. And some examples would be mad cow disease, the ability of this. So with mad cow disease, remember that's prions, those proteins that get folded incorrectly. And remember how stable they are. It's very difficult to deactivate them. And so if they become in a cattle population, and if you've learned about how cattle are farmed these days, are usually put into huge feedlots where they are jammed together, and mainly just dirt. They don't have any pasture. It's just soil and dirt, really, and they're commingling. And remember, prions don't break down. It takes a heck of a lot to get them to break down and become inactive. And so the modern farming practices are promoting that. Also, I think I might have mentioned, if we looked at prions, they had to come up with new laws on how to butcher meat because of prions. And the prions can end up in the nerve tissue. So they had to be much more careful about how they get rid of the nerve tissue when they butcher. Lyme disease, we do have that around here. Heavy population of Lyme out east, especially, but we do have it around here. And again, it's this idea of the ticks, the way we are encroaching into different population or areas where we, cut down forests and so forth and build subdivisions. So we're encroaching into their habitats and we're getting more exposure. Transfusions and transplants has, unfortunately, before they caught it, hepatitis C was being spread that way. Live animal markets in various areas of the world help spread disease as well. And then E. coli 0157H7, you've heard about that one. That one has caused problems multiple times. And this is looking at primarily what ends up being one of the biggest problems is when the cattle are killed and then they're sent to the meat processing plants. The way the meat is handled can cause this E. coli to end up in the meat that is then sold to us. And I'll talk more about that when we get into food microbiology. So again, just thinking about some of the behavior patterns that we have, or demographics that can influence what we see in terms of ease of transmission and getting exposed to new, emerging or re-emerging pathogens. And then don't forget that microbes are very quick to adapt. They have the ability to mutate and adapt much more quickly than we do. They can get that access to genes they've never had before through horizontal gene transfer. And so there's all that dynamic going on as well. I'll mention it in the food. Okay. Then we need to think about climate change. And certainly that they're studying this pretty carefully, trying to figure out how we can try to mitigate some of these issues in terms of with climate change. They're starting to see that you can have the emergence of new pathogens through the process of climate change. And some that they've already documented pretty nicely when they look at mountainous regions where people are found, that because of the air temperature is getting warmer, that's allowing some of the vectors to survive and go further up in altitude and can get altitude in higher latitudes. And so that can affect populations of people living in those areas that never really had to worry about that in the not too distant past. So we're seeing this they say it's extending the range of the pathogen and allowing for the pathogen to be more successful in areas that previously it just couldn't survive. With climate change, there's areas of the world that are getting higher rates of rain and flooding. And of course, mosquitoes need water. They need standing water to complete their life cycle. And that is certainly another easy way for the mosquito population to explode and get more reach out into the community more so than ever. So with all of the increasing knowledge that they're gaining in the field of epidemiology, they're starting to propose this idea that's gaining traction. It's called the One Health Initiative. The One Health Initiative. So they're trying to take a more holistic look at how do we keep populations of humans. Of course, we're very centered in on humans, but not just humans. Globally, we're looking at all life forms, but looking at the pathogen host interaction and the idea that we have to think about all these other aspects. We have to think about all the vectors, we have to think about all the reservoirs, all of it. We really need an interdisciplinary joint effort in order to tackle this idea. And I think this has gained more traction since the pandemic because that was a big, wake-up call that we really don't know as much as we think, and being able to make very quick decisions on information that is lacking is a tricky business. So looking at human health, but also other animals, and looking at insects and vectors and all of that. So they're even, saying, well, we could take steps like we could vaccinate rodents to decrease Lyme disease. That would be a strategy we could employ. And so again, this is an interdisciplinary effort to get those people out into the areas where they can do that. Certainly plants as well, looking at growing plants in a safe way. I'll talk to you a little bit more about this in the food section next, but getting plant pathologists involved, because now more than ever, we're realizing that a lot of human pathogens can be kind of hidden away in foods, in plants that get harvested and then sold off for food. So we'll talk more about this, but basically, it was kind of a wake-up call. They started realizing that some human pathogens can survive inside plants. And so if you're a microbe hanging out inside plants, the fancy word is endophyte. If you're an endophyte, that's what you do. You hang out in plants and you don't really cause the plant any harm. In fact, and a lot of endophytes tend to help the plant. But in this case, it's a very specific case. We're talking about human pathogens that can get into the plant, survive, tucked away safely in the plant. And then when you eat them, the chance that you could get sick from that. And so they're starting to look at this more carefully when they realize that initially the assumption was human pathogens probably die off in the plant if they get on the plant or something like that. If you wash off the plant, you're good. And now they're saying not only do you have to wash your produce, and that's a good idea, but that doesn't mean it's going to solve the problem. You can't really get rid of the endophytes. There's no wash you can do to your food to get rid of the endophytes. And so that's a whole nother level of scary that they can be lurking inside the plant. So far, there hasn't been a lot of documented transmission with this method, but it's something that they're concerned about for sure. Okay, so the One Health Initiative, you'll probably hear more about that. It's a big, it started amongst microbiologists, but now it's starting to spread out into medical doctors and other people in other areas of discipline. Okay. All right, so that's what I'm going to cover with the epidemiology idea. So between what we talked about here and then the bit that you did in lab, hopefully you have a better understanding of epidemiology and some of those concepts. The next topic I want to get into is food. Is that in EduCat? Is that, you don't see it there? Okay, I can get that. Okay, if you refresh, it should show up now. Is it there? Okay. Let me share again. Whoops. Hmm. All right, I gotta stop share. It just gave me a message. No, no. It said something weird about my audio, but it looks like it's working. Okay. So now, with food microbiology, basically the topic I'm going to focus on is looking at how to keep yourself safe from eating food that's contaminated. And the first several slides I'm not going to cover. It's more for you to just read. It's actually the cover of a book. So I scanned, this is a book over in the library. But it has some quotes in there that I thought were really interesting and quite relevant to the idea of how when you eat food and drink drinks of water or whatever, that's the most intimate interaction you have with your environment because you're taking something from your environment and putting it internally into you, and then your cells and microbes are interacting with it. So that when you think about it from that perspective, it's a really good idea to understand what you're eating, what you're drinking, and to be a little bit more careful about, we have to worry not only about our own agricultural system, but when you look at packages in the grocery store, it's amazing how many packages are labeled, grown in Mexico, grown in Peru, grown in wherever. I mean, it's all over the globe that we're getting these foods. And so, do you know anything about their agricultural practices? How they work through with pesticides and chemicals? What kind of food handling laws do they have in place? So there's all kinds of things to think about and just ponder. And then I wanted to point out there's two terms here, food spoilage and food contamination, both caused by microbes. And the one I'm going to be more focused on is food contamination because that is more of a hidden idea. The microbes are hiding, they're not so obvious, and then you don't realize they're there, and then you eat or drink that and can get ill. Food spoilage, on the other hand, we're talking about if you leave something out for too long or in the refrigerator too long, eventually the microbes that are on that food will start to degrade the food to such an extent that you can tell it's spoiled, right? I'm sure you've done that before. Have you ever had sometimes I don't eat toast very much, and so jam gets shoved to the back of the fridge. Have you ever had that where you open the jar and there's like a velvet covering of fungi over the surface? Quite shocking. It's like this carpet of fungi. But there's all kinds of ideas of food spoilage. I'm not gonna really talk too much about it. Like, I'm not gonna ask these bullets here. I'm not really gonna ask you to memorize. That's not the point of this. I'm putting some of the detail here on food spoilage, just so you're aware of it for your own life and as you eat food and prepare food. But if you're curious of why, sometimes you take that swig of milk before you realize it's spoiled. I'm sure you had pretty nasty taste biting something. So those are usually fairly obvious either by sight or smell. And then if you happen to be the unlucky person who tastes it, you certainly can taste it. Food contamination, though, is human pathogens lurking in or on the food, and you don't really know they're there, is the main point. And so then you can get exposed. So food spoilage, again, I'm not going to ask you to really study this for the final exam. This is more just for your own knowledge. In case you ever wondered why the milk went sour, you can have an appreciation now that you've gone through this class of how the microbes are altering the food. And again, from their perspective, they're just eating the food for their own nutrition. They're not, that's their goal. But in the process, they can spoil the food. And so this, again, the food spoilage tables is just for your own knowledge. And this is another one where they kind of break it down into categories of which bacteria and which fungi tend to be dominant for these issues. This is an exhaustive list. There's others out there, but these are the heavy hitters that show up frequently. So even high sugar foods, like I mentioned, jam, jelly, you can still get contamination, both by bacteria and fungi. Some of them can be quite deadly. This one, okay, so this slide, I do want you to know. All right, so food spoilage. When you are looking at the different foods you have in your kitchen or pantry area, it has to do a lot with how much moisture is in that food in terms of the likelihood of that food spoiling. And so you can break it down into perishable, semi-perishable, non-perishable based on moisture. That's how they do it. Water content. So they actually can measure the so-called water activity, A sub W, and they have tables of all the foodstuffs and what their water activity is. And so the way to interpret that is that pure water is set at 1. That's how that's the reference. And then some examples, it'll never go above 1. Okay, so that's pure water. But salted fish, for example, we have a water activity of 0.75. And then chocolate, honey, dried milk, 0.6. So the lower the number, the less water in that food. And the longer it should stay fresh and not spoiled. So the table there, I think you could easily come up with some of these ideas on your own in terms of what's perishable versus not perishable. So pay attention to this slide. Okay, and then the rest of the slides moving forward, I think, are all fair game. So going back to the plant idea, I wanted to talk a little bit about that because they're getting more research being done on this, looking at just in general, looking at the plant microbiome. Yeah. Many botany people are very, very interested in that for a variety of reasons. For this topic, though, I'm zooming in on this idea that you can get microbes hanging out either in or on plants that could be negatively impacting humans once you get exposed to those plants. So this image is just a big picture idea. It's saying, okay, when you look at a plant, what are we thinking about for the microbiome? For us, we zoomed in a lot on the intestines, but remember, you have a bunch of microbes on you as well. Same with plants. They have a bunch of microbes on their leaf surface, their stems, their roots. But they can also have microbes inside them everywhere. Okay, so they can be inside the root tissue, stem tissue, leaf tissue, even flower tissue, seeds, fruit. So they're everywhere. And like I said, most of the microbes that hang out with plants, they don't care about you. And in fact, if you get exposed to them, they may actually be good for you. So I'm not really thinking too much about that. It's more of what happens if you get the wrong microbes in or out. So I thought that picture on the top left was quite nice, where they're showing a leaf, and they have I think they sprayed it with Serratia and then they let it sit for a while and then they imprinted it onto the auger just to see. I think they were doing a timed experiment, like how long can Serratia sit on a leaf before it might die. And so when they took that time, obviously it was very, very healthy. For those of you who have not studied any kind of plants, this image on the top right, that's the leaf surface, highly magnified, where they're showing the core of the leaf, which is called the stomata. Stomata in particular. So that's the opening there that is taking in CO2 and giving off oxygen, and all the purple are bacteria. So you can see the size scale difference. You can see how easy it is physically for microbes, bacteria, to crawl into the, so to speak, crawl into the pore of the leaf. And so leaves have pores all over their surface, these stomata, and they have to have them open in order to take in the food. They can close them though if they have to, and they can. So there's nice studies showing where the bacteria are gaining entrance into the stomata, and they actually react to that. The plants react to that and end up closing their stomata, at least temporarily, to try to fight the infection. So then here, this lower left image is just looking at, in general, when you think about endophytes with plants, their definition is that they don't harm the plant and may actually be helping the plant. And there's a variety of ways they can help. But we also have to consider there could be endophytes getting in, there could be bacteria, fungi getting in to the plant and existing in as an endophyte, but they might not be so healthy for us. And that includes also when we eat seeds and nuts, because those are not sterile. Even if you wash the surface, there are endophytes in the seeds as well. So they're all parts of the plant. And of course, we eat lots of plants. So here, this image is looking at the seed idea, and they are tracking how endophytes get into the seed as the seed is forming. They know it can happen. And so, seeds and nuts can be very nutritious, but they're not sterile. Here's a nice visual showing the different groups of microbes that can be found in different parts of the plant. This I'm just showing you to give you that appreciation that there's a lot of diversity with the different microbes. Phyllosphere, that top word in the box there, phyllosphere, that's microbes that live on the leaf primarily. We're talking about hanging out on top of the leaf or on the leaf. And then the rest of the they zoom in on endophytes for a lot of that, but they also get into bulk soil. So the reason that they're also studying soil, field soil, obviously, but most endophytes they think come from the soil environment. And so understanding what microbes are in the soil and how we manipulate the soil as we grow plants, for example, by adding manure and other things like that, could we be creating problems in terms of how we manipulate the soil? Could we be introducing things into the soil that maybe could end up in the plant and then we get exposed to it and eat the plant? Right, so big picture idea here, looking at not just the plant, but also what we're doing to the soil environment as we grow these plants. So this paper is looking at manure fertilization and its impacts on human pathogens that are found inside vegetables. So the endosphere is in them. So there's no cleaning when you bring that vegetable home and try to clean it, surface clean it. There's nothing you can do about the endophytes. They're there. You're not going to get rid of them. Whoops. So then, whoops, what happened there? There. So this is a picture taken from one of the figures from one of the papers where they're saying, yeah, we need to be thoughtful about this. And there are some areas where people are using human feces as fertilizer. There was at one point, and of course I'm exhausted, but so I may be off on this, but I thought even at one point the city of Ishpeming was looking at this idea they weren't saying human feces. They phrased it as biosolids, and they were looking at a biosolid like manufacturing plant. I don't know. Does that ring a bell with anybody? I don't think it came to be yet, but uh, there are other places where they use human feces. They process it. They have to process it a certain way. Hopefully it's processed correctly so that we're not putting human pathogens into the soil. But that's something I'm sure they're looking at to make sure that the pathogens aren't getting spread. So when you have food contamination, it's implying you can't tell that the food's contaminated. You can't see it, smell it, taste it, and yet you ingest it or drink it, and it causes illness in you. So the CDC estimates, and this is I think a very low estimate, 76 million cases of gastrointestinal illnesses each year due to contaminated. And again, that I think is really a low estimate because most people who get sick from food don't go to the doctor and it never gets reported. And so most of the time we just, try to survive the misery and then go about it back to our life. We never go to the doctor about it. So that's roughly 1 in 4 Americans each year. Probably one of the better studied ones was salmonella in, unfortunately, peanut products, peanut butter. So this is some of the data from that. And you can see the kind of the bell-shaped curve here of the different across the timeline. Definitely peaking and then finally subsided. And then they actually started using some nice gene sequencing to be able to figure out where it was coming from, what lot of peanut butter was the kind of like what company was selling it. Then so this information you can find, on the CDC's website. You can look at these types of alerts that they'll post. They're pretty good at doing it right away, and you can do that and find out. I'll show you in a minute here soon. So salmonella, they didn't know it was in the peanut butter. No difference in taste or smell or sight of the food. And yet it was there. So salmonella is kind of tricky because once it's in the food processing plant, it can persist. If they don't have good hygiene practices in the plant, you can see that in this image, they did dry surfaces, which dry surfaces are usually the same. Microbes have a harder time surviving on a dry surface because all living organisms need water. And yet when they swab and sampled the dry surfaces in that food processing plant, they could show that salmonella was very happy and living and viable. So different microbes have different tolerances to dry areas. Salmonella is pretty good at hanging out. So we have different ways. A lot of it is with our agricultural practices and including manure addition, both with raising food like fruits and vegetables, as well as for meat production. So no facet of agriculture is safe if they have poor practices. Listeria monocytogenes is a very serious pathogen that's gaining a lot of scrutiny again. There's a couple that are really making people nervous because Listeria, well, you gotta kind of admire this one because it is so tough. It can survive in your freezer and actually slowly multiply. It is totally fine in your refrigerator and happily multiplies. And you can't tell that it's in the food on the food. You have no idea. This one is very dangerous if you are ever choosing to become pregnant. You have to be very aware of this pathogen. luckily my doctor, when I was pregnant, she was totally up on it. But the sad thing she told me, she said, I can't have ice cream. Tell a pregnant person you can't have ice cream? I mean, come on, crazy. But then she put me in. So she said, no ice cream. She even tried to tell me not to do chocolate. That I couldn't give up. I was just very, very careful about what kind of chocolate, source of chocolate. And then things like she would tell me it's very happy to live in like on meat. And so if you go to the deli and you buy, sliced deli meat, that's one that's risky. And so she said, if you do that, or even if you buy the packaged meat on the grocery shelf, she said you should microwave the meat until it's like um, steaming to kill. At least you can kill it that way. And so you have to be very, very careful on how you prepare your food because it's so happy in so many different foods. There was a famous case study of it over in France. It was on cheese. There was a wine and cheese party, and it was all over the cheese, and a bunch of people got really, really sick. So it can make you really sick, and then it can cause miscarriages. And if the baby does survive, if the mom gets really sick, could cause could cause health problems in the child if it survives it. So really a heavy hitter. So it's a psychophile. So be careful about that one. And if you're immunocompromised, that's the other one. If you're immunocompromised in any way, it can really hit you hard. Okay, I think we'll stop there. We'll get that next topic is going to take a while. So we'll continue on with that on Friday.